Okay, thank you, Julianne, and good morning, everyone, and welcome to the second lecture in the series Innovation Gainesville, where we're exploring our world-renowned innovation sector. Last week, we heard from Mark Long, the director of UF Innovate, who talked to us about how ideas become commercial enterprises through the help of UF Innovate's incubator system. And today, I'd like to introduce Dr. Barry Byrne from UF's College of Medicine, who will be speaking to us on the future of genetic medicine and his work at Lacerta Therapeutics at UF Innovate's Sid Martin Biotech, where he is a co-founder and scientific advisor. Dr. Byrne received his MD Ph.D. degrees in microbiology and immunology at the University of Illinois. And after a pediatric residency, cardiology fellowship, and postdoctoral training in biology chemistry at Johns Hopkins, he joined the University of Florida in 1997, where he's currently the professor and associate chair of pediatrics and director of the Powell Gene Therapy Center and Child Health Research Institute. He's also the Earl and Christy Powell University Chair in Genetics at UF's College of Medicine. As a clinician scientist, Dr. Byrne studies a variety of rare diseases with the specific goal of developing therapies for inherited muscle disease, with a focus on conditions that lead to skeletal muscle weakness and abnormalities in heart and respiratory function. His group has made significant contributions to the understanding and treatment of Pompe disease, which is an inherited disorder resulting in a type of neuromuscular disease caused by excess glycogen storage. To accomplish this, his research team has been developing new therapies using adeno-associated virus, or AAV, mediated gene therapy, which is a process by which genes can be introduced into humans and some primates on a small attenuated virus to correct genetic mutations. Dr. Byrne is using this specific approach to restore cardiac and skeletal muscle function in Duchenne muscular dystrophy and Friedreich's ataxia, as well as Pompe and other inherited neuromuscular diseases. So with that, I'd like to welcome Dr. Barry Byrne.
Thanks very much. I appreciate you having me to join Innovate Gainesville. And I'm going to talk, as was said in the introduction, about the work we've been doing over the last few years since I joined the University of Florida, where really this technology was developed, in fact, 30 years ago. And so I'll give you a little bit of that history and why we're focusing on these issues now. So really over the past 10 years, there's been a continuous revolution going on very quietly. That is the genomics era that we are now in, where genetic tests can establish the fundamental basis of a disease. But unlike what we saw here in 2012, when this cover of Time magazine said these tests may point to risk, but not always a cure, we're now in an era where the therapeutic opportunities that are derived from knowing the genetic basis of disease can establish a new class of drugs, genetic medicines that treat the specific underlying cause of a problem. And I'll give you a few examples of inherited recessive diseases that are rare, but these same principles apply to many common conditions like Alzheimer's disease, Parkinson's disease, and even potentially diabetes. So let me start by giving you a little further emphasis of why in pediatric care this is so important, because among the worldwide population, about 10% have a rare disease, or over 400 million people. So any one type of condition is itself rare, but collectively they're in fact quite common. And half of those are children. Sadly, about a third of those people with rare diseases who don't survive the first year of life, and that's a problem that we face in one of the most common conditions. And particularly in Florida, we see about one child every week with Duchenne muscular dystrophy. So 50 or so patients born per year with this inherited muscle disease that is ultimately fatal in the late teen and early adult years. So how can we address this? Well, conventional medicines have been tried, but not very successful in this area. So we really turn to the DNA that causes the disease. So this is a somewhat of a schematic idea that you could encapsulate genes into a pill. But in fact, what was invented here at UF 30 years ago by Ken Burns, who was for a period the head of the Genetics Institute and dean of the College of Medicine, and his collaborators at UF, was a way to put genes into a small virus. And this is a picture of that virus that was beautifully mapped out by Mavis Abanji McKenna and her husband, Robert. Sadly, last week, Mavis passed away from ALS. And so, but we have a legacy really of discovery, and her commitment to teaching and research was just phenomenal. So we really will miss Mavis, but will always be remembered by these images she generated by solving the structure of AAV and other parvoviruses. So this is a unique virus that was not much attention given to early on, because the virus doesn't really cause any independent disease. It's associated with or it co-infects individuals alongside of a common pathogen, adenovirus, that causes respiratory illnesses. But it was isolated from some infections in a daycare setting, and all those years ago, and Dr. Burns and the team imagined how this could be used to not deliver its own genes, which is what the wild type virus carries, but therapeutic genes. And so the ability of AAV to now reach all the cells in the body, particularly in the heart and the brain, has led to a genetic medicine revolution that is part of what I described, taking advantage of all the knowledge we've gained about causes of genetically defined disease. We first got involved in this area when I was still in Baltimore and published this paper in 1996, which is now the basis for a whole series of studies done in muscle. And over the course of the 5 years following this, soon after I came to UF, this finding was replicated by a number of groups, and we found that we could get persistence of gene expression, in this case of a gene that makes a protein to just mark the muscle or color it, in this case by a stain that we see here in this blue color. But we saw that there was a functional aspect of this, because we could express the protein that causes increase in red blood cell number in varying amounts. So 3 different groups here, here, and here. The higher the amount, the dose of the vector, the higher the level of erythropoietin expression. And that was led to a patent that we had on this topic to demonstrate that AAV vectors could be used to deliver therapeutic proteins in muscle. And in fact, that has led to these many years later, a study that I'll tell you about in the end. So how did all this come about? Well, Dr. Burns and his group really first isolated the AAV genome way back in the early 70s. And then a professor in microbiology, Nick Maziska, conceived of the idea in the early 80s that those viruses could be used to deliver therapeutic genes as vectors. Dr. Burns' group advanced the idea that these vectors could, in fact, and the virus itself could live in cells long-term through integration. And then when I came and several groups began working on the therapeutic strategies using AAV, we continued the work on muscle transduction. Bill Hausworth's team focused on retinal delivery of AAV for a variety of forms of inherited blindness. And we had a real innovation from Sergei Zolotukhin to express a marker, a different marker gene that was less toxic to cells, that could track the expression pattern in the brain and other tissues. And this is called green fluorescent protein. And I'll show you an example of that as well. And then we started the clinical program. So late 96, we founded the Gene Therapy Center with the help of Earl and Christy Powell's charitable gift to the center, and initially working on alpha-1 antitrypsin deficiency. And I know you're going to have the opportunity to hear from Dr. Brantley at some point. One of the first trials that was done was for alpha-1 antitrypsin deficiency here called AAT. This was done with Dr. Flawed and Dr. Brantley. Then early and mid 2000s, the work began on the inherited retinal diseases. And the first one that was chosen was Leber's congenital amaurosis, or LCA. And LCA is a deficiency of a retinal pigmented epithelium protein, the RPE65, that leads to a retinal dystrophy and blindness in adulthood. Although the observations can be made early in childhood, and I'll talk more about this in a moment about what was the result of those clinical studies done initially at UF. And then we initiated a study in a form of Parkinson's disease known as aromatic amino acid decarboxylase deficiency, which was one of the first trials to test the AAV vectors delivered into the brain directly. The work done on muscular dystrophy started a number of years later, and I'll explain how that's contributing to the notion of delivering these AAV vectors throughout the body. And for all of this, we needed to manufacture enough of these products to actually make them viable commercial drugs. So what will be the consequence of this long term is that we see here the number of early stage companies that start off out in this far circle, which are called phase 1-2 trials to establish safety. And as one gets closer and closer to the center, where pivotal studies are done to prove the effects are drug related, versus those that reach the very center where marketed products are established. And here's Luxturna, which is the drug that was the first approval of an AAV gene therapy based on the work of Dr. Hausworth's group for RPE65 deficiency. So ultimately, as more and more of these programs reach the center of the bullseye here, this will really change clinical practice. And this brings up the idea, are we finding the right drug that exists to give to the right patient in a personalized approach versus individualized medicine, which means creating from scratch the right drug for a given condition. And that, of course, means knowing that a given disease is caused by a single gene abnormality. And this is an example for in ALS, for example, and about 20% of ALS patients have inherited an abnormality in a specific gene that's so-called familial ALS. But we know for many of the early onset diseases that are seen in children, these are conditions in which both parents are the carriers of the abnormal gene, and then about a quarter of the children can be affected. And one of the best examples of that, at least that reached the popular media and press, is the inspiring story by the Crowley family. And here's John and Eileen Crowley and their two children, Megan and Patrick, who are affected by Pompe disease, and their older son, John Jr., who's there behind Patrick. And they really inspired the story behind a movie called Extraordinary Measures, which I was fortunate enough to participate in, and in the way that story was told. And I'll show, there are not many medical movies that have such an inspiring story, but the story became told by Brendan Fraser and Harrison Ford. And here's a brief clip from the movie that described the dilemma faced in the period when this movie came out. There were no effective therapies for Pompe disease. And here's one of the parts of the story that was told to the family, played by Brendan Fraser during the movie.
After all, it's not just her lungs, okay? Her heart, her liver, these organs have been compromised and would have become fatally enlarged. Now, I wish that we had a drug to treat Pompe, but we simply don't.
So he says, I wish we had a drug to treat Pompe, but we simply don't. And of course, that too often was the message we had to deliver patients who were faced with these severe conditions, which were, as I said, half the time fatal. So how do we go about solving the problem? And this is what's now known as translational medicine, or in this project development timeline here, we begin with a research program. This often extends far to the left, where there are many, many years of research. And in fact, we've been working on a Pompe gene therapy study for the past 15 years. That leads to early studies in animals that are viewed by the FDA as proof of concept. These are prior to the clinic or preclinical testing. During that whole multi-year period, these are, at least a 5-year timeline here, but it often lasts longer than that. You start to make the clinical material, and I'll talk a little bit about what's been done at UF to contribute to that knowledge. And then this is the period in which, once that material is manufactured, an investigational new drug, or IND, is submitted to the FDA. Once that process is done, it enables the agency to approve an early phase clinical trial. And we're in the process of doing that in a number of circumstances now that I'll describe. And here's what's required to manufacture the material. We have to have controlled storage of the material. We follow a specific process for purifying the materials on these fancy machines called a chromatography instrument. And then operators who work in the GMP space work inside of a biosafety cabinet, just like other pharmaceuticals are manufactured. And we do that all here on campus in the McKnight Brain Institute. This is a nearly 2,000 square foot facility that makes both cell therapy products and viral products. There are 2 production suites rooms within each suite. And then there's an additional facility in the Cancer and Genetics Research Complex that works almost all on cell therapy products. Here's an example again of 3 operators in their gowned environment working on the manufacturing process side by side in the biosafety cabinet and processing the materials for future human use. This is a very complex process. It's like any pharmaceutical manufacturing done to the highest standards. And so we were really one of the first to establish a facility like this in an academic setting. This requires adhering to a set of federal guidelines that we now, from the time this was created, now have almost 1,000 controlled documents to provide the specifications of how the work is done. And that requires a quality group that we have in place. And then once all that's done, the information is submitted to the FDA. We go to the Institutional Review Board, which governs the patient safety in clinical research, and seek their approval. And then we work with the study sponsor and our principal investigators here in the Academic Health Center to develop an investigative plan. And we describe all of the components of that plan, as well as how the product was made and the preclinical study reports. and that leads to a clinical trial. So just more schematically, the animal-related work is all prior to the clinic. That involves making the clinical product and testing its toxicology or the maximally tolerated dose. And then we take all those findings into that facility I just showed you and establish the manufacturing and release of the product. We've done now 8 of these studies over the course of the years, and with varying amounts of the product used. But this, the current era now allows us to not use these in just the eye, for example. So here's the RPE65 product that was the second one, two forms of the alpha-1 antitrypsin, and then the RPE65 product was done to treat about a dozen patients with inherited blindness. And this was ultimately led to the very first marketing approval of a gene therapy drug in the US called Luxturna. So this is what Luxturna looks like. It is about $800,000 to receive this treatment. And this is a very small amount of the vector is injected behind the pigmented epithelium, behind the retina, next to the pigmented epithelium, which is at the very back of the eye. And so this was in 2017. I was able to serve on the FDA advisory committee that led to this approval, and it was based on the work done here at UF and ultimately licensed by Spark Therapeutics, a company in Philadelphia formed from the Children's Hospital Philadelphia. And now this is, for these past 4 years, been commercially available to all patients with Leber's congenital amaurosis. But making a very small amount of the product was relatively easy, frankly. The amount of material required for thousands of eye patients may only be enough for one individual that needs systemic therapy. So I'll give you an example of how we have tried to capitalize on knowledge about how to make AAV to move us to the next level for systemic therapies or adult therapies. So Dr. Marks of the FDA really thinks about manufacturing capacity and safety of the clinical product in this sense, like a Starbucks. These little ones are easier to get. There's a lot of this middle size made, but it's really hard to make a lot, and we need new technology to make this viable. The enabling of the large-scale manufacturing was done as part of a collaboration. Soon after Luxturna was approved, we started work with a company called Solid Biosciences, whose founders had a son with Duchenne muscular dystrophy, so that problem I mentioned at the very beginning. And what was necessary was to determine how to grow the AAV vectors in a way that could be used for hundreds of patients and not just one at a time. And so we took advantage of what was established in the industry to some degrees for certain proteins, but not for viral vectors, the ability to grow large-scale cultures up to 200 or even 2,000 liters in what's called a bioreactor. And we now, won't say this is routine, but we now have really a better acceptance of this methodology as the way to make AAV vectors that enables their use in lots of patients. And what we learned from the work at Solid was that if we, this is an example of that green staining I mentioned, but for a different protein, for dystrophin, which is present around every muscle membrane to improve the integrity of muscle. And here is in a mouse, the normal staining for dystrophin in the heart, the diaphragm, the muscle at the front of the leg, and the thigh muscle. And in a MDX mouse that has not been treated, you see we don't find any of that staining. But using the drug that was developed for Duchenne muscular dystrophy, we found very robust staining in the heart, the diaphragm, the leg muscle, and the thigh muscle that was really encouraging. And that led to a clinical trial, which we've just completed the first portion of. And here's some of the learnings from 9 of the patients in the study. Red is not good. It represents decline. And so there were 3 control patients who were untreated here. And you can see that in breathing function and in walking function, they all had a decline. There were mixed results in the group that received a low dose of the material, some improvements, and in some other aspects, a decline. But in all the patients treated in the high dose group, they had improvements in almost every category. Reduction in the marker that indicates the integrity of the muscle membrane, improvements in walk distance, and improvements in vital capacity, as well as the a quality of life measure called the PODC, Sports and Global Measurement. So this was very encouraging. This will allow us to continue this study and use this dose, which was determined to be effective in additional patients. And in fact, next week, we have another patient enrolled in that study, the 8th one overall. So how will this whole process change clinical practice? As studies move from this outer phase 1-2 area back into the target zone of approval, sorry, it keeps jumping. We expect that early phase studies will rapidly lead to marketing approval, and the FDA expects up to 40 marketing applications in the next 5 years. So this will accelerate very quickly. Most of these are for early onset inherited diseases for which there's no other treatment paradigm. And we've used this same technology to treat single patients that have a very unique condition which may be amenable to only 1 or 2 patients. And it's really important that we improve access for rare disease populations as a means to help drive the innovation and make these therapies available even for the smallest patient communities that might be out there. And some of the enabling of that comes from confidence in the financial side of it. This is where commercialization is required to really have these therapies be sustainable. In 2019, there were almost $10 billion investment in this sector, and nearly $8 billion of it in the gene therapy arena, and still significant contributions in both tissue engineering and cell therapies, where they share a lot of common features. Another metric for the interest in the topic has been the commitment of large pharma to enter into this area, which traditionally was only the risk profile was only allowed for biotech companies or early stage companies to be involved. And now we see Astellas, Roche, Vertex, Biogen, Bayer, all major pharmaceutical companies making investments, in some case multi-billion-dollar investments, to advance these therapies and make them accessible to wider patient populations, both in the US and outside the US. And here's a few examples. A treatment for thalassemia was approved by Bluebird Bio. Then beyond Luxturna, the next treatment of real significance for our patient community in pediatrics is a treatment for type 1 spinal muscular atrophy. And the treatment was developed by Avexis, which one of our programs contributed a lot to the work done at Nationwide Children's in Columbus. That was the starting point for Avexis. And I'll show you at the end, we did the first commercial infusion of this Zolgensma therapy and are now working hard to make it available to wider groups of patients through some research we're doing with Novartis. And then Novartis developed Kymriah, which is a different kind of virus, but used in a form of inherited cancer. And here's what's additional highlights of that. Valrox is a treatment for severe hemophilia. There is a program for a form of leukodystrophy that was filed for in Europe. And then there are a lot of cell therapy programs which we know have been very impactful in the oncology space. And we work closely with our colleagues in neuro-oncology to try to help advance those programs as well. And you can see here in the bottom right the number of anticipated programs last year and into next year. So very, very encouraging pipeline of products that are coming along. And I mentioned that we did this first commercial infusion of Zolgensma at UF Health now almost a year ago. This baby was not identified through newborn screening, so had needed some support, both feeding and respiratory support, but is now doing quite well. And in fact, she has an affected brother who was identified through the newborn screening program that was implemented in the state several months ago. So how will we manage these cases in the future across the country? So I have the privilege of being the Chief Medical Advisor for the Muscular Dystrophy Association, and the MDA has about 2,000 providers across the country, where we see over 90,000 patients a year, and at 150 care centers, with a lot of these patients participating in clinical trials. And so this initiative is called Innovations in Care. It's partnered with the Innovations in Research effort at the MDA, where early stage grants are provided to investigators in academia. So just end by thanking the people within our center that support this work in translational medicine, Dr. Manuela Corti, in working with our business colleagues, Kirsten Coleman, and the running of the Human Applications Lab is Brian Cleaver, and our early stage vector core by Sanford Boy, who is one of the key innovators of the LCA program done in Bill Hausworth group, and then the quality insurance group. And just end by showing you how much it really takes a village to do all these studies. So this is our clinical and research team involved in the Duchenne program I mentioned with Solid Biosciences, both laboratory staff and clinical research staff from all aspects of the program, from the very earliest discovery research through a clinical implementation. So I'll end there by saying the Global Genes program has this motto that hope is in our genes, and that's certainly for families that don't have access to other therapies. This kind of innovative medical therapy has been really, really rewarding and helping them see the future for their children. So we can end there and take questions.
Okay, well, thank you so much, Dr. Byrne. That was a terrific lecture. It's an amazing amount of work that you've done. And I find genetic therapies fascinating. I guess my main question for you would be, what do you see as the biggest hurdle in solving genetic diseases? So, I mean, why don't we have more diseases resolved at this
Well,
point?
The,
What are
the
the
process for the early stage studies to be completed involves a very lengthy program to establish the greatest level of safety and scrutiny of how the product's made. So we put a lot of effort into that. We see the opportunity for really linking our discovery research efforts on campus with the efforts at the Innovation Hub, working with the team at UF Innovate to build on the commercialization aspect. Of course, that involves carefully balancing our institutional and individual conflicts, and that's managed through disclosure and a very detailed attention to that aspect, and that enables us to move those programs along, manage those conflicts, and see that really the goal of getting these therapies to patients is what's achieved in the end.
Okay, well, so some genetic disorders are more complicated than others, where you have multi-gene involvement versus maybe a single gene involvement. So where do we stand with that balance? Are we able to use this AAV approach to deliver genes in diseases where so for instance, I think of cystic fibrosis. I know we have a lot of different variations.
Fibrosis was one of the more favored programs for implementation of gene therapy, but it turned out to be one of the most complicated because of the environment in the CF lung was very challenging for getting persistent expression of genes. But there are opportunities for multi-gene problems to be managed in the same way. We just need to know how to combine those parts that are relevant. And I think one of the big challenges we face is how do we switch genes on and off like is normally done in the body? We don't have a good way to do that currently. But the group may have read about gene editing. That's a really an interesting opportunity which allows the control that existed within a given cell to continue to be active, but to repair the part of the gene that might have a defect. So, and that gene editing actually uses the same AAV technology that we've developed for rare diseases.
Mm-hmm. Well, I probably have a dozen more questions I could ask you, but I think let's open it up, Julianne, to...
I see Shirley, go ahead if you want to unmute yourself.
Hi, Dr. Byrne. This is so interesting. I wonder, what is your thinking about that doctor in China? Was he an explorer? Was he a criminal? What do you think about it?
Very, very good question. So what was done in China was to use a gene editing technology in an embryo that ultimately gave rise to a birth. Though this was an alteration of the germline of this patient, but in a strange way to make that individual resistant to HIV infection. Did not seem to make a lot of sense, frankly. This was the susceptibility to HIV is not a fatal problem that needed to be corrected in that individual. It was just a demonstration of how gene editing could be used in the germ cell. And that's one area where I think scientists generally agree there's too much of an ethical dilemma there to consider that a reasonable medical therapy. And so I think there's general agreement in the scientific community that experiment violated ethical and general safety considerations, because there could have, even though it appears to have been safe, there could have been very significant untoward consequences. And that, of course, you have to keep your mind on the first principle of do no harm. And even though there was, no harm to that individual that was born, I think it crossed an ethical boundary that people feel need to be legislated against. So that was probably not a good idea. And
Do you think
I
it's
don't
the
think
wave
you'll
of the
see it be
future?
done again.
Do you think it's the wave of the future?
I don't, not in somatic cells that could change the heritability of a trait. Fetal gene therapy in a fatal condition that would otherwise receive, not be viable is another consideration. And there have been thoughts about that. That's different because those traits are not passed on. So whether it's prenatal or after birth probably doesn't matter a lot but may impact the feasibility. So say a condition has very untoward consequences from the day of birth onward, it might be conceivable that if a few weeks or even months before birth, that infant might be treated. And that's a whole different story than what was done in that experiment in China. So, but right now, essentially, we do have neonatal treatment of SMA because of newborn screening. And there are about 35 conditions now that are screened in the general population at the time of birth, some of which have very severe consequences if not treated early. And one of those is SMA, for which there's now a gene therapy treatment. So we'll see, I think, more treatments like that. Our topic that affects the Crowley family for Pompeii is also on newborn screening. And we foresee in the next year the ability to treat those infants as well. The only current therapy is a repeated visits to receive an infusion therapy that's given every 2 weeks. And we just did an event with the FDA where patients talked about their experience and quality of life on these kinds of treatments. And as an example of the impact on that on a child who might not be symptomatic until their early adult years would have undergone 3 years of hospital-based treatments if you begin the treatment as an infant because of its repeated nature. If it's a gene therapy strategy where the treatment is only given once, something I should emphasize, the unique nature of these treatments is they're generally only given once in a lifetime, then that obviously has a big benefit for patients who might otherwise be exposed to multiple doctor's visits to receive a treatment. So we think that's a potential advantage. Other questions?
I do have
I
one.
think, go ahead, Julian.
Well, I was going to read the one from chat, but do you want to do that, Paula? Is that what you were
Yeah,
going to do?
sure. It's from Eli Glazer. His comment is, across all DNA-based clinical programs, where are the funding sources in the US and worldwide?
Yeah, so in the US, we've had over the past 20 years, tremendous federal support for the research programs that lead to the early discovery. The government, though, is not in a position to seek marketing approval and make these drugs sustainable for patient access. That's really up to the pharmaceutical industry. So there's a mixture of both federal and private funding. The funding, as I mentioned, is coming now from pharmaceutical companies, from venture capital in early stage companies, and from biopharma companies that are kind of in between the two. So we do think there's still a unique circumstance where foundations and philanthropy will contribute to enabling these treatments to be available for really small populations that for which they may not be commercially viable. That's still, I think, a big challenge that we face. And so we haven't solved that problem yet, other than the notion that these might be done through a nonprofit mechanism or something in that arena, because the large pharmaceutical companies are not going to pursue the really ultra orphan programs where there may be only a handful of individuals who are affected.
Shirley, did you have another question?
This is so interesting. I was wondering, when you do your clinical trials, Dr. Byrne, do you do them like normally? Is there 2 arms to it? Do you have controls and
That's a great question. Initially, that was not the case. They were all what is known as open label studies where everyone received the treatment and possibly different amounts of the treatment, different dose levels. But the agency has recommended now, the FDA has recommended, in order to improve the certainty that the treatment has the intended effect, and because drugs need to be both safe and effective, they have moved to recommending placebo-controlled trials and where a group of the participants don't get therapy, at least initially. And these are also called delayed start trials, in which the control group has to wait a period of time, often a year, to gain access to the treatment. And then one compares the treated to the untreated group. And in that example I showed from solid, there was a control group that declined compared to the treated group that had improvements.
Fascinating.
I had just, I guess, an extension of my earlier question. So how do you decide that you're going to work on, say, Pompe disease or some of the genetic eye diseases? What are the criteria kind of go into?
Well, one of my teachers long ago said you have to start with a good idea. So it means understanding how the genetic defect causes the condition. So we have to know the mechanism. And in some cases, we know there's a gene associated with a disease, but we don't know how it causes disease, exactly what process happens in the body over what period of time. In the case of SMA, there were animal models that had the genetic defect that was similar to patients. And then the treatment was found to dramatically change the survival of the animals. So that's pretty clear cut when you have survival versus not. The treatment rescues the early death of the animals. That was the basis by which the SMA program went forward. And that's similar in our Pompe studies. We have early mortality in the untreated animals and then survival in the treated animals.
Mhm.
And and in there'll be some circumstances where there isn't an animal model that accurately predicts the course of the condition. And in that case, we may only be able to test the therapy in humans. And this is where adult trials, so there's a natural disconnect though, in that more advanced cases in older individuals may not be as amenable to the treatment, but only in adults can we get the proper informed consent to conduct a study that may not have a true therapeutic advantage. And in children, the study is required if there's any risk associated, which is true of all gene therapy studies. The study can't proceed unless there's some potential for benefit in the individual. And so we have to balance those two kind of opposing views. And often that means that a set of the patient's initial group might be adults who can do the proper informed consent. They may not have the maximum benefit, but then it would allow the younger subjects to be subsequently enrolled in the study. So yeah, it really starts with fully understanding the condition and the potential for reversibility. This is important. If we know that there are accumulated problems that can't be corrected with the gene therapy, those may not be the best candidates. And that's why newborn screening, early intervention, all helps to contribute to more successful treatments in this arena.
Well, and I think I heard you mention early on that you were looking at type 1 diabetes, which of course we do have a good animal model for, but which is a really complicated autoimmune disease. So is that something that you're headed toward?
I do think that, most of the hospitalizations in diabetes are related to the complete absence of insulin production. So we do think there could be an opportunity for avoiding that condition or diabetic ketoacidosis by getting some sustained expression, low level expression of insulin. Plus, we now have some innovative ways to control gene expression that might be responsive to feeding, so or postprandial glucose. So if we had a way to monitor glucose levels in the blood, which exists now continuously, it may be possible to regulate the gene expression in response to that. And that's a project we're very interested in working on with the group in the Diabetes Institute.
Are you working with Todd Briscoe by any chance, or?
Yeah.
Oh, okay. He's going to be speaking to us next week. So,
Oh, good.
yeah. Sure. I'm sorry, Shirley, you had a question.
I think I heard you mention that newborns are now screened for 34 different.
Yeah, 35 different
illnesses.
conditions.
Are there potential treatments for all of those? Is that why?
That's a very also excellent question. It's a requirement of qualifying for newborn screening recommendation. So there are no screen conditions for which there's no therapy, because it's a significant commitment of resources to do the screening. So we have to make sure that there'll be some benefit to undergoing the screening. And it's an interesting process because there are federal recommendations, but states must implement them. And Florida is one of a few states that once there is a federal recommendation, we have 18 months to decide up or down on whether to proceed with screening. So the SMA screening decision went very quickly, but it actually took us a whole year and a half in that committee to contemplate and enact the Pompe screening, because there's additional complexity in that one, because we will actually identify adults with Pompe disease who will be throughout most of their early childhood and adult life asymptomatic, but identified as children. Of course, this changes the way their parents then interact with them on the anticipating what's going to happen later in life. That required a lot of consideration of how the counseling would be done for those families. And we're still learning how to properly deliver that message so it doesn't create too much concern on behalf of those patients.
Are these a simple blood test to the newborn, like the
Yes,
PK?
yeah, they come from the blood spot card, which is collected from the heel stick in every newborn. There are some screening tests that are physical tests. There's one for congenital hearing abnormalities, and there's another test that I participated in developing while I was still in Baltimore that uses you've all ever had this red light on your fingertip, the oximeter that measures blood oxygen levels. That's now used to screen for congenital heart disease in babies.
Wow. Oh, fascinating.
Very, very interesting. Thank you so much.
So we've got one final question here from Eli. He's got an edgy question for you. When and should we provide every newborn with a full DNA record for lifetime use in healthcare? And how is the cost for a DNA profile projected?
Also an outstanding question. It is the premise of a movie. In fact, if you all want to catch up on an old movie called Gattaca with Ethan Hawke and Uma Thurman, this was the principle, and this movie was from 1990, before there was DNA sequencing widely available. So it was very futuristic idea that one's whole life course would be influenced by their DNA. And parents had the ability to choose whether they would have a natural birth or one that was free of any diseases. And this is the story about two brothers, one of whom is engineered and the other's not. And it really is an interesting story about self-determination, because what happens in the movie is that the one with the anticipated genetic problems actually overcomes all of those limitations and does live his life's dream. But this is now commonplace in some countries. In Canada, for example, newborn whole exome sequencing is done routinely for those that are hospitalized or begin on a new medical therapy to try to anticipate drug reactions rather than through trial and error. There is a whole field known as pharmacogenomics, and this is now actually routinely done for people who receive blood thinners. So there are certain levels of susceptibility to adverse reactions to blood thinners that are tested by very specific gene sequencing, as well as other medications which were known to work in one population versus another. That genetic testing exists. But in theory now, the costs are, because of the technological advances in this genomic era we're in, the costs are such that it is just as is said here, this could be available to everyone. And in fact, the NIH initiated a program to provide sequencing data to a large cohort of individuals. And one could imagine that in part, it was kind of ironic that in the COVID pandemic that the US, which led the way in developing sequencing technology, was in the very middle of the pack in sequencing of all countries, of sequencing the genome of coronavirus, because no one anticipated how the virus might be changing, how its genetic composition was influencing these variants that are observed now. So we have really amped up the infrastructure for sequencing, both of all of these, the whole population whose so many screening tests are being done for COVID. That same technology applies to human genetic sequencing as well. And now the cost of sequencing a genome is about $200. Now that's much more than a newborn screening test. Newborn screening tests are only a few dollars per test. And so what one can imagine really exactly is said in the question, maybe it's better to just sequence a whole lot of things at once and then pick and choose which ones out of that information should be reported. Trying to think of the name of the reporter. One interesting story, I think it's Charles Kuralt, whose family had Huntington's disease. And he tells a very interesting story about how, frankly, as a young adult, he wouldn't have wanted to know that he had this condition, which would have affected him in his later life, because it does influence your thinking about your capability. So it's a tricky business to decode the whole genome and make predictions about one's future health. But certainly when there's a known early risk, this approach would be very successful. So that's where we stand with it. There's some ethical dilemmas there, but in terms of informed consent and when and what to tell an individual who has this sequencing technology, but it's now commonly used in families. Anyone who's signed up for any of the ancestry programs kind of is familiar. How many people have family members who've participated in any kind of sequencing or family heritage study? Yeah, no, it's fast. It's fast. I have. It's fascinating. I made a gift to our kids to do this, and so the various forms, whether it's Ancestry or Google, they provide a lot of information about not just ancestry, but also disease susceptibility, and they're learning more all the time, and will theoretically go back to some of those susceptibility genes and look more carefully at other genes to see whether it's predictive of any serious medical problems.
Are we getting a complete genome sequence with those,
I think when those things were started, they are only snippets, if you will, of the genome that help establish certain patterns of migration and inheritance. So there are gene patterns that are seen in different regions of the world that can be traced back to the DNA. And there's some fascinating stories in those kind of population-based studies about how people migrated out of Africa. Some who survived cold climates went to the north. Others who did not have the genetic predisposition to survive the winter went to the south. So all those that came over the Alaskan land bridge into North America were cold tolerant people. And that's been learned from looking at their genetic footprints and their origins out of Europe.
Okay, well, I don't think I see any more questions. So I want to thank Dr. Barry Byrne tremendously for a great lecture today and interesting discussion afterwards. We very much appreciate your time with us today. And as I mentioned, Dr. Todd Brusco will be talking to us next week on human immune system development and looking at genetic variability and susceptibility to type 1 diabetes. So hope you all can join us then.
Thank you all for your support of this series, and thanks to UF Innovate for hosting, and to Mark Long. So appreciate it.
Thank you all.
Bye
See you
now.
next week.
Thank you.
Stay well.